All right, guys, let's quickly go through this so we can get going over that lab and then also starting the next one. Okay, so what cellular transport process is most responsible for helping pull water up a plant? Oh, not reverse, that's artificial. Just osmosis. Okay, reverse osmosis is entirely artificial process. Okay, so yeah, we got to have just osmosis here. Okay. For number 2, where in the stem would you find the phloem? Outside. Okay, what gases are stored in the spongy layer? Carbon dioxide and oxygen. Okay, if they have water vapor there, I would say also accept that. So we'll say 1 mark each for those. Okay, 1 mark for oxygen, 1 mark for CO2. If they have water there, okay, they're only going to get 2 marks for this question if they have O2 and water or CO2 and water or whatever. Okay, it's 2 marks if they have some combination of those 3. Okay. All right, for number 4, what was the major advantage of multicellularity? Specialization. Okay, that was the big one. Number 5, what's the difference between a true multicellular organism and a colony of independent cells? Right. Okay. In fact, we can say division of labor if they have that. See? So DOL, division of labor. Or you can also write exactly the same thing you wrote for the last answer. Okay. Specialization. All right. I would accept that also as the as an answer for that one here. Hang on, guys. Okay. For number 6 here, what cells in the plant change their shape the most by changing the amount of water in their vacuoles? Anyone remember? Nope. Yeah, I'll accept either stomates, but I was looking specifically for the cells that make up the stomate. Okay, the guard cells. All right, I'll accept either one of those as the answer there. Just know in the future, guys, I'm going to be looking for the actual name of the cells. That's guard cells. 2 guard cells make up a stomate. Okay. Number 7, the tubes that conduct water up a tree are called? Nope. Xylem. Okay, what are the 2 functions of the stem? Does 2 things. That's one of them. Okay, conducts, because I gave it to you in the question before. Okay, and the other one, support. Conduction and support. 1 mark each for those. No, because that's what the roots do. Okay, number 9. As days get shorter, a plant would secrete more of this stuff to signal dormancy. Well, yeah, if you remember it that way, that's a good way to remember it. Abscisic acid. All right, and number 10, what substance secreted by plants acts as a growth hormone, lengthening the plant? I'll accept gibberellins, but I was actually looking for auxins. Okay, gibberellins do cause lengthening of the plant, but the primary growth hormone are auxin. Okay. All right, so that is out of 12. Give him a mark out of 12. Because there were 2 questions that were out of 2. Okay, so mark out of 12, let them see it. Put it in the box on top of those literacy assignments. Okay, so we're going to have to move pretty quickly here because we got about 25 minutes left, but setting up this lab doesn't take long at all. All right, what we want to do is, okay, go over there and essentially cut up our pieces of plant, okay, and get them in a beaker of dyed water, all right, or sorry, not a beaker, a graduated cylinder of dyed water, so that they can photosynthesize and transport water over the weekend, and then on Monday we can see how much water they have moved. Yeah, Julianne? Grayson, thank you. All right, so Our manipulated, responding, controlled variables for this lab. First off, the manipulated variable, because it's hard to guess this one, okay? But the manipulated variable here is this. We are manipulating how the plant can transport water. Okay? Some people say plants transport water by pumping it from their roots, and others know the right answer. Okay? They transport it using osmotic pulling force from above in the leaves. So what we're doing is we're cutting off pieces of the plant. What is that eliminating as a source of force to transport water? The roots, right? We're cutting off, we're just keeping the leaves. If they're not attached to the roots anymore and it can still transport water, then the roots have nothing to do with it. Agreed? Okay, so we're manipulating essentially the structure of the plant, okay, or the way it can transport water. We're saying you can only transport it using the thing I think transports water. Responding variable. What are we probably going to watch for? Well, we hope that that's going to go on because that's what's going to cause the water to move. But the only way we'll know that's happened is changing of color of the leaf and changing of volume of the water. Okay? Hopefully there'll be less water in there on Monday. Okay? Controlled variables. Okay? In there, you need to list anything you think can affect how well a plant transports water and also carries out photosynthesis. All right, so there's some factors in there that affect how well a plant can carry out photosynthesis, and as a result, how much water it will transport. Those are our controlled variables. Those you need to think of. 3. Okay, they're mostly environmental. All right, for our hypothesis, we want to generate a hypothesis about what forces are involved in the transport of water in plants. So our if part, okay, should probably talk about what forces we think are responsible. Okay, and as always, describe the experiment. Okay, then tell me what you expect to see. All right, now, I don't have the experimental apparatus set up here like it says. Good, I've got pictures of it from last semester. Okay, so what we're going to do here, and it's very important that you follow these instructions to the letter so that this experiment works. Okay, what you are going to do is, I'll have the plants in the sink, okay, at the front of the room. Or actually, I'll probably have them in a couple of the lab stations instead, because the front table's got chemicals on it. Okay, so we'll put them in 2 sinks. What you need to do is cut a piece of that plant off. Okay, and take it back. Now, that's actually, sorry, that's not the first thing you're going to do. You're going to grab a graduated cylinder. All right, graduated cylinders look like this. Okay, you're going to need a couple of them for this reason. Okay, hopefully you'll grab like a fairly large or not a fairly large, a moderate sized graduated cylinder. Okay, and you'll grab a big one. Everybody with me so far? And grab like the 100 ml one because that's where you're going to mix and measure. In the big one, fill it till it reads 100 mils. Don't fill it to the top because then you won't know how much you have in there because you'll go past where the measurements are. Everybody with me? Okay, put it in there. Put like 2 drops, maybe 3 at the most, of food coloring into the water. You don't need any more than that. Okay, it'll be plenty dark enough with just that many. Okay, stir it up, mix it up, okay, so that it's all blended, and then you're going to pour it into the smaller graduated cylinder, and you're going to fill that graduated cylinder very close to the top. Don't fill it all the way to the top, because then when you put your plant in, some will spill out. All right, so fill it to, let's say, this close to the top. All right, everybody with me so far? Now, how are you going to know how much water you just poured in there? Right. Okay. You see how much has gone from here. You go 100 minus whatever's left in this graduated cylinder, and that will be your initial volume of water. That is a number you have to record. Okay. We need to know how much water we start with. So 100 minus whatever's left in there is your initial volume of water. On Monday, there will be less water in there. All right. So once you've got the graduated cylinder full of water, okay, you're going to come up to me, and I'll have some rubber gloves at the front of the room. Okay, we're going to use the rubber gloves to seal the top of the graduated cylinder. Okay, so you come up, and I'm going to give you the finger off of a glove to put over your graduated cylinder. Okay, now once you put that over the graduated cylinder, you need to poke a hole in the top, a small hole. Okay, this is how we're trying to seal the plant against the top so we don't have a lot of evaporation coming out of the graduated cylinder itself. We're trying to hope here that all the evaporation will be out of the plant. You guys follow me there? So just poke a small hole. If you have a pencil, that's the best thing. Poke a small hole with the pencil, okay, and then you'll come up and you'll cut off a piece of the plant. When you're cutting off the plant, it is important that you cut it underwater. Okay, so take the plant and just take it underwater and cut it off. Then put your thumb over the cut end. We don't want any water, or sorry, we don't want any air to get into the xylem tubes. If it does, it disrupts the hydrogen bonds and the cohesion and adhesion that goes on in there, and we don't want that to happen. So put your thumb over the end, okay? Go back to your setup, to your lab station where you have your graduated cylinder with the finger over the top, okay? And then quickly take your thumb off and put it in there. Okay, everybody with me? Now you got to do it quick. We don't want to get any air in there. When it's all said and done, it should look something like this. And notice how deep, okay, that plant goes into the water. You got to make sure it goes deep enough in there that it won't be out of the water by Monday. All right, everybody follow? So you got to cut off a fair amount below the leaves you get. Okay, you're probably going to want about 2 leaves. Okay. If you got 2 leaves, you're probably good for a number of leaves. Okay, and make sure you've got a section of stem below those leaves that'll go nice and deep into that graduated cylinder. All right, once you have this, I'll give you a piece of masking tape, write your names on it, put it around the graduated cylinder, put it on the windowsill, we're done. Okay? Everybody good with that? All right, so again, the key thing here, okay, is when you cut that plant, that you cut it underwater like that terrible picture shows, okay? You can see here she's got 3 leaves on the plant that she used. Right, 2 will be lots for us. Okay, yeah, pretty straightforward. What do you need to record today? You need to record the initial volume of water and take a picture of your plant. I would say take close-up pictures of the leaves because we want to see if there's any changes in the leaves by Monday. All right, so I don't have, yeah, so okay, you can see here, here's kind of a before and after shot, okay, of what happens in this lab. Okay, you can see here that this one has got a lot of food coloring that's moved into it. Okay, and this one here has, okay, is the sort of start of it, and it's not very green. Okay, here's the start of it here too. Okay, again, not really green in there to start with. Or sorry, very green to start with. What food coloring color should you not use? Or yellow? You wouldn't believe how many people do that if I don't tell them not to. And then they come to me on Monday and go, Mr. Couture, nothing happened. You're right. Nothing happened. Go sit down. Okay? Use the blue and the red. If you want to mix blue and red, it makes purple. It's very pretty. Go ahead ahead. Okay? Doesn't bother me. Just don't use green or yellow or this lab will be a total flop for you. Okay? All right. Let's head across to the chem lab, guys. A little bit here about the analysis and interpretation. Okay, number 1 is going to be done on Monday. Okay, when you subtract, okay, the initial volume you had today and the final volume you have then. Okay, and then you'll be able to compare that to other people. Okay, describe the flow of water through the plant. Okay, basically you're going to refer to your notes. Okay, to answer this one, as well as refer to how your observations tell you that that's what happened. Okay, predict how leaving the roots on would affect the volume of water flowing through the plants. Okay, so that's one you think about and tell me what you think would happen. Okay, what if we added salt to the water? For this one, think about what that would do to that process. Okay, to osmosis. How would that affect osmosis if the water was salt water, not fresh water? Okay. Why did you place the plant in the sunlight? How would the results been different if you'd have kept it in the dark? Okay, think about what does a plant need in order to survive, stuff like that. Okay, if you place the entire plant and its leaves underwater, what would happen? Okay, and number 6, discuss the natural reactions and adaptations of plants that might help maintain their water balance. We talked about those. Okay, we said there's like 3. Okay, Jenna? Oh, sorry, yeah, these two here are under class discussion now, aren't they? 5 and 6. No, no. Just do the first. Yeah. Just do the 3 that are on there. All right. And then conclusion. Remember to do it. Okay. Restate your hypothesis, and then tell me about how do your results tell you that the plant was able to transport water or not. Okay. And you'll have your pictures that you took today and tomorrow. Or today, hopefully. Did everyone take a picture of their leaf today? Okay. If you didn't, go back in there and take one before you go.